by Brian Wiegmann. Genomic data supports a new phylogeny-enabled identification system for Anastrepha. Brian.
Well, good morning. As you can see, we've been working hard in the laboratory, basically taking this incredible and exciting biodiversity and trying to build data sets that will reveal to us patterns in their evolutionary history. And I'm going to talk to you about a project, the results of a project that's been going on for about 5 years that includes Alan Norbaum and Raul Ruiz and Gary Steck and a large group of people who have been working together to really integrate multiple data sets across the study of Anastrepha to get identification tools available so that these fruit pests basically can be identified rapidly. And we have the opportunity to sort of bring next-gen sequencing data to those questions. So as you know, Anastrepha is the largest genus of Tephritidae in the New World, over 300 species, and many undescribed as well that Alan and Gary and others have been working on bringing some sense to this really amazing radiation of New World species. The larvae feed on fruits or seeds, and 10 or more species are serious pests, and they attack fruits. And we've been working on basically trying to integrate multiple data sets on these questions. This work builds on the really exciting work published not that long ago by Chimo Mengual and Alan Norbaum, in which they analyzed multiple genes to produce this rather comprehensive molecular phylogeny of Anastrepha, in which multiple groups were established, which would be the Anastrepha species groups, the various groups, to bring some sort of organization to the diversity that has been accumulating from the intensive efforts in field work across the Central and South American tropics, and trying to bring a modern sense to the classification of Anastrepha. So there are 28 species groups recognized in Mengual in Alan's work, but you can see the diversity of species within these groups. There's also been a rather good amount of work in population genomics showing that the species groups are difficult taxonomic problem because there's genomic integration going on in these. So some of these are definitely in the process of speciation and have not yet sort of become fully isolated. So genetic signals have been studied in laboratory of Conganes and Reno, and this work is basically confirming that the complications of understanding the taxonomic classification of Anastrepha is really due to a complicated genetic history, and that we shouldn't expect this to be a simple problem. Nevertheless, it's very important to agriculture and commerce and the commerce in fruit around the world. And so we would like to reconstruct the phylogenetic relationships with Anastrepha using hundreds of nuclear markers, and then use those nuclear loci as identification tools for the pest species and the cryptic species. So that would lead to tests of markers for rapid and real-time identification by PCR. My role in this has been to sort of use our phylogenomic data reconstruction to build essentially a phylogenetically informed database of nuclear loci so that we can sample from that database for those markers. You've heard all about it here. We use anchored hybrid enrichment and a pipeline of analysis tools in order to go from raw read DNA libraries all the way through orthology search to get those specific single copy nuclear orthologs, and then we're doing standard phylogenetics. And so what this is leading to, all right, if I'm lost on the slides, what this has led to now is an unparalleledly, if you want, large phylogeny of Anastrepha that, like the previous talks, right, it's very hard to tell you about in its entirety. I have the tree here with the branch lengths turned into a sort of a normalized as a cladogram there so that you can see 740 specimens on here. This is about 150 species, and this particular tree is 275 orthologous loci present in 90% of the taxa. If I were to show you the tree with branch lengths, it becomes extremely difficult. Anyway, it's the one over there on the right. So this is a very rapid radiation of many genetic samples. What I can tell you just in general is that across this tree, we have strong concordance with the species groups, previously established species groups. There's much support within the tree with high support values for the monophyly of species clusters throughout Anastrepha. We're very excited to publish very soon an analysis of the interrelationships of these species groups. Of course, there are large parts of the phylogeny, like the Mucronota species group, for example, which is para or polyphyletic and needs more taxonomic work, and you'll hear a little bit more about that in a later talk by Eric Rodriguez. What's perplexing and difficult for all people studying Anastrepha is the Fraterculus species group, a large radiation, and we have many trees and many analyses designed to try to uncover the phylogenetic signal for the history of the Fraterculus group's radiation. And so we're seeing patterns of monophyly within there, as well as sort of interesting new clades and patterns of perhaps introgression in the tips of some of these clades that make it a species complex that's extremely difficult to resolve. So what we're doing with this now is taking this phylogenetic framework and 275 nuclear loci up to about 2,000 nuclear loci that we have for Anastrepha, and we can build a queryable, a searchable phylogenetic reference system. And to do that, we've started to use this program called TBAS, the phylogenetic tree viewer, which is developed by a colleague of mine at NC State University for Ascomycetes phylogeny. When anybody sequences a new Ascomycete, they can submit that sequence. It gets realigned with the data that exists. An analysis goes out to the San Diego Supercomputing Center through Cypress and comes back with a tree and a placement. So we're working on developing this system to do real-time phylogenetic placement of unknowns, but also to publish a reference system for Anastrepha that can be queried and searched with respect to things like a mitochondrial CO1 sequence or an ITS sequence. And so just to demonstrate how that works, here's a smaller tree of Anastrepha species, a test system, if you will, showing the relationships among groups. The green group around the edge here is the Fraterculus group, for example. And so we can use this system to investigate, zoom in and out of small parts of the tree, and then basically look at individual loci and their information content with respect to parts of that tree. And so as we use that tree as a reference system, each taxon is connected to its metadata like any database, right? And we can start to sort of reveal patterns in the geographic distribution, in the populational history, and then also in the evolution of these nuclear loci. This is a challenging problem. And just because we have the ability to collect an enormous amount of nucleotide data, making some sense of it is going to keep us busy for quite some time. And so this is an example of that TBAS interface. What we're doing now is testing for the information content in various nuclear markers. And we want to compare that to the standard identification tools that have been used, like CO1 barcodes, which we've got over several thousand of them now for all of the same samples, as well as ITS1 and ITS2, using this reference system. We started that process by using a program called PhyDesign, which looks at the rate of evolution of individual loci and sort of allows you to see relative to that tree that's in the upper right-hand corner of this slide, relative to that tree, you can see what genes are evolving at an increased rate within the tips of that tree. That allows us to sort of pare down loci to look for those with particular information content in the tree. And that's just sort of a very general overview. But we would necessarily perhaps look at those loci individually. So this is a 100 taxon tree for which we have CO1 nuclear, 600 nuclear genes and ITS. And you can see that loci differ in both their taxon coverage, in their conservation, and in whether there are informative sites within them. And so any of us familiar with sort of the search for synapomorphies across our groups, this is completely identical to that problem, right? So nothing has changed with respect to how difficult systematics is in a highly biodiverse group. We would like to find perfect synapomorphies as identification tools for these pest groups. We have the bioinformatic tools to help us organize that search, and we're working on that now. And there are many bioinformatic tools that we're working on. So for example, new methods for phylogenetic placement with very small amounts of DNA material have been designed because of the COVID pandemic, for example. There are incredible methods for using phylogenetic placement to quickly place where you are in a phylogenetic tree. So essentially, we will use SNPs and SNP analysis, where this anchored hybrid enrichment data set is equivalent to a genome-wide sort of survey of genetic variation in there. And together with graduate student Escher Cattle, we are now going through locus by locus using bioinformatic tools to sort of design probes that will serve as, for example, here, an identification tool for placing an unknown taxon in the tree. So I think that's about it. This is where we're headed: multiple overlapping phylogenetic views of Anastrepha diversity and using nuclear genes to help us get marker systems that will work. Thanks.
Can you go back to the slide and show the alignment?
Right.
So some of those are not very satisfactory.
Mm-hmm.
I guess you include that for a reason to show how that helps.
Right. So I think we all know this. That one on the blue one there is definitely something you would want to get rid of, right? I guess my point would be that we get an enormous amount of information. You're going to hear in other talks probably tomorrow or whatever that not all of the loci in our phylogenomic surveys are good phylogenomic or phylogenetic markers. Not all of them would be good identification tools there. And so we have a couple of choices. We can go locus by locus through thousands of loci and throw some out and keep others. But what we're really doing is trying to use bioinformatic tools to whittle down the search to specific identifiable markers. Thanks.
Thank you.